good afternoon, everyone. Welcome to HBCSE, and we are very happy to have with us Aswin from NCBS. He's an associate professor at NCBS since 2010, and he is interested in his lab works on various aspects of bacterial regulatory networks, genome organization, and transcription factor evolution since that time. If you're wondering about his background, he's actually an engineer. He did his BTech in industrial biotechnology from Anna University in Chennai. And following that, he went to EMBL to do his PhD, and also in University of Cambridge with that was a postdoc, I think. Same, okay, with Nick Luscombe. And he seems to have a really long list of things he's interested in outside of science, such as he's a Carnatic music singer. He's interested in watercolors, analog photography, reading fiction and pop history, and this is my favorite, I think, collecting and using handmade fountain pens. So anyway, we are very happy to welcome him. He'll be visiting HBCSE for a couple of weeks, and I hope that you have a chance to interact with him. Thank you so much. Please welcome.
Thanks, Mayuri, for hosting me here, and thank you for coming to this talk. Metabolic plasticity, and how we've come to appreciate their metabolic plasticity over the last 100 years or so, and maybe towards the end talk a little bit about the kinds of work we do in the lab, and how that fits in with the metabolic plasticity of bacteria. And there are also nice things that can be done fairly easily in an undergrad setting, or probably even at home if you have a computer, and internet, which these days everybody has.
Is the mic on?
It's here. Oh. Just a second. But hello. Is it better? Is it better? Ah, great. Done. Okay. Right. This is a photo that is taken in February or March this year. That's the season that passes off as autumn in Bengaluru. And see, this is the pond in NCBS, and it's a nice lily pond. And there's a lot of these leaves that have fallen. There's the lily leaves, and then there are all these dry leaves that have fallen here. And all of these, if they're not cleared, and they get cleared here, they're going to be broken down by the microbes that live in this water. And as they say, this is going to severely increase the BOD of the water, and that could eventually lead to problems for other kinds of organisms living on that water. So this is essentially a microbial process. So I'll start off with some numbers. So here we have the population of humans on the planet. It's about 10 to the 10, close to 10 to the 10 now, and we just keep scrolling, and then somewhere here at around 10 to the 18, we get one more number, and that's the insect population on this planet. Quite a large number, and then it just scrolls down, and the number of bacteria and archaea, or prokaryotes as we call them together, is estimated to be somewhere in the 10 to the 30. And this is a very, very large number, and what do we call it? I don't know. And these bacteria live pretty much any habitat that you think of on the planet, you have these organisms, and they're probably most commonly found in the subsurface, both terrestrial and oceanic, and even in things like the Arctic ice caps, there are like 10 to the 24, 25 bacteria that are estimated to live there. Now, if we look at the phylogenetic space, that's the tree of life. The bacteria here and a couple of other groups, eukaryotes, which includes us, and archaea, they're all here, and the good chunk of the phylogenetic space at the moment that we know of is taken up by bacteria. And to the extent that some 50 years ago, no, well, 70 years ago, it's only recently that we're calling 1950s as 50 years ago. Well, 70 years ago, Cliver and Van Niel, in this very, very nice little book, which is a collection of lectures called The Microbes' Contribution to Something or the Other, little red book, say, without microbes, only a few human individuals, by taking very special measures, would be able to lead a highly artificial and charmless life. And we can think of those prominent individuals who are thinking of living in Mars or whatever it is these days. Right, so we started learning about bacteria about 350 years ago when Antony von Leeuwenhoek first saw them under the microscope, and since the mid 19th century or so, our knowledge of these organisms has grown in many different different ways. We won't get into all of that, but I'll focus on one aspect, that is the metabolic plasticity that I mentioned. So possibly the first systematic study that I've come across that is looked at bacterial adaptability and its ability to change its properties is this 1907 paper by Frederick Twort. Frederick Twort seems to have been a fairly interesting person. Is most famous as the discoverer of the bacteriophage, which he did in 1915. But he seems to have had many interesting traits. I think he was a spectacular microbiologist from what he had done, and apparently he could also make clocks, he made violins, and he could imitate a chimpanzee. So he had many such interesting traits, and the institution, unfortunately, that he worked in, called the Brown Institution, was bombed during the war, so it doesn't exist anymore in London. So in this 1907 study, he took some bacteria which were incapable of metabolizing a certain type set of sugars, and then he grew them in the lab in the presence of that sugar until they developed the ability to start metabolizing these sugars. So today, a lot of people do these laboratory evolution experiments where we take bacteria, put them in a certain condition, and keep growing them for generation after generation till we find variants that are better suited to the condition in which we are growing these bacteria. And nowadays we do that, and then we immediately sequence whatever comes out and see what is the genetic basis of the adaptation that we find. This was probably the earliest such study, at least the earliest in my knowledge. So a bacterium which did not have the ability to metabolize some nutrient X gains that ability over a matter of some tens of generations. And then this study, which is more famous as the study that led eventually to the discovery of DNA as the genetic material, was also an example of metabolic plasticity. Metabolic plasticity defined in a very broad sense. So where you have a bacteria which are incapable of causing disease mixed with related bacteria capable of causing disease but heat killed, suddenly develop the ability to cause a disease, the famous Griffith experiment. Well, there's a lot of fine detail to this work. This paper runs to 47 pages or something like that, and it's very dense. And Griffith, I don't think he really knew what he had discovered in terms of its impact on our understanding of inheritance and so on, but others did thankfully, and that went somewhere. But here, this is an example of bacterium not having a certain ability within a very short period of time discovering this new thing. And similarly, 20 years later, 25 years later, two major papers came out discovering conjugation and transduction, which is essentially mechanisms by which bacteria gain new genetic material from other bacteria or through bacteriophages, and therefore by doing so change their traits from state A to state B. And then the very famous Luria-Delbruck experiment. In retrospect, when we look at all of these are mechanisms by which bacteria gain new DNA from outside and thus develop a new trait. But here, Luria and Delbruck looked at just the process of mutation, and all of this was 1943. Every MacLeod study had not yet been published, so we didn't yet know that DNA was the genetic material, leave alone Watson and Crick type stuff. So here they looked at mutations that confer resistance to bacteriophage, and then asked whether these mutations are pre-existing in the population and then get selected, as Darwin would have said, or whether a certain condition that has been posed produce these mutations. And these are mutations which convert bacteria that are susceptible to killing by bacteriophages into something that's resistant to the same bacteriophages. So more examples of the ability to adapt. And though we now talk quite a bit about AMR and its impact on health and so on, this was already appreciated as early as in 1920s to the extent that I can see. The first person to sort of talk about this, again first in my knowledge, was Alexander Fleming himself, and this was before the discovery of penicillin. Fleming also discovered lysozyme, and when in his working on lysozyme, he did identify that certain colonies of this particular bacterium that he had used to study lysozyme could gain resistance to this to the killing action of this enzyme. And again, some time later, this was around the time sulfonamide drugs had been introduced, and several labs, including Fleming himself, did various experiments to show that bacteria initially susceptible to these sulfonamides can, when continuously exposed to these drugs, gain the ability to resist these same antibiotics. And certain such epidemics of resistant bacteria started spreading during the war, the Great War of the Second World War, and some people said that it is just providence that penicillin came out at that time. Otherwise, it could have had a serious problem on our hands. And talking about penicillin, 1940, before penicillin's large-scale introduction had happened, Chain and Abraham, who were involved in the large-scale synthesis of penicillin, discovered penicillinase, the enzyme that breaks down penicillin, and whose relatives are all our carbapenemase and all of that, NDM1, blah, blah, blah. All of these are relatives of these things. Maybe not entirely, depending on which family of beta-lactamase you're talking about. Okay, so enter the 1960s, there were quite a few things we knew. So we had quite a bit of knowledge of their metabolism, their funny metabolism that we cannot do, essentially be chemolithotrophs, that is make use of inorganic sources of nutrition. We now knew what their genetic material is, and apparently there were still people who wouldn't believe that DNA was the genetic material, but by then the weight of evidence was so heavy that if you thought otherwise, then prove it. The DNA guys didn't have to prove anything to anybody. That they're highly metabolically flexible and adaptable was also known, and we had some understanding of their ecology and their role in biogeochemical cycles. We knew about their role in fermentation 100 years before this, and so on. But what we didn't know was we didn't know how to define a bacterium. And in this article, Stanier and Van Niel sort of said, any good biologist finds it intellectually distressing to devote his life to the study of a group that cannot be readily and satisfactorily defined. And they call it the abiding intellectual scandal of bacteriology at this time, is that they don't have a systematic definition of bacteria. And when we're talking about defining an organism, that also to some extent means classifying them in some way. And for organisms, classification also becomes genealogical as opposed to an artificial classification of books in a library. And this genealogical classification, which is also called phylogenetics, a term that was coined by Ernst Haeckel, is what Charles Darwin talked about. I have an ancestor, and then from there I have bifurcating tree-like things, and then various descendants come, traits transfer along this tree in various ways. And Ernst Haeckel also had this idea where he put this particular thing called Monera, some simple organism was the base, and then plants, animals, and protists were three branches that came out of this base. And there are this whole huge number of these lovely gnarled oak tree drawings that one can get from Ernst Haeckel's work. Now, in this sort of a tree of life, where do you really put a bacteria? And if you're going back to this time, this is the mid-19th century, people still believed in spontaneous generation. When Louis Pasteur showed that fermentation was a product of yeast, it was not as if nobody knew about yeast. They knew about yeast, but they thought they were the products of fermentation, and not that fermentation was product of yeast. So where do you start putting these organisms? Are these organisms? People are seeing these things under the microscope. They're seeing different properties, shapes, pigmentation, motility, etc., etc. But are all these different forms you see, are these different forms of the same thing, or do you still have some sort of a species definition where a trait that you see is heritable over time? Ferdinand Kohn was one of these people who did think about this. He had he did classify various bacteria, and then he said, okay, I can classify them based on certain properties. For example, I'll divide them into X number of groups based on the shapes that I observe, and then further subdivide them based on, for example, their biochemical properties and so on. And is this a library-like classification, or is it a phylogenetic classification? I believe that he did see this as a phylogenetic. They decided that they will have some kind of a tree, and they said, no, these spherical cocci would be at the base, and then something will happen, and various other clades of bacteria would emerge from this kind of an ancestor. 20 years later, when they wrote about defining a bacterium, they said, well, we no longer defend this hypothesis. So it's and instead they say, okay, but we still think that it is possible to define a bacterium, because if a microbiologist looks at a bacterium and a eukaryotic microbe under a microscope, they'll be able to tell, okay, this is a bacterium, this is not. But ask them to systematically define, how they came up, to know. So, but the fact that they would through their experience and all that, tells that there is a particular pragmatic basis to define the bacteria. And then they went on to say that, okay, I will define it based on cell properties again, cytologically. I'll call all these unicellular organisms as prokaryotes or eukaryotes. Quoting an Edward Shatton, who had published these names a little earlier, and using this, he could say that blue-green algae, which are cyanobacteria, are bacteria, and that protozoa, fungi, are not bacteria; they are eukaryotes. And is this again a phylogenetic definition? They're thinking, okay, the microbial world has probably diversified along two lineages. One is the prokaryote lineage, and one is the eukaryote lineage. So if that is how the tree of life is, then yeah, this can be phylogenetic. If prokaryotic cells and eukaryotic cells are two monophyletic clades on a phylogenetic tree, then no, this cytological definition is also a phylogenetic definition. fair enough. But is that true? So 1960s, again a little later, Linus Pauling and Zuckerlund, they came up with this idea. So all along you've been trying to classify organisms using various morphological properties, but the morphological properties are not the thing on which evolution really acts. But of course, all along we didn't know how to access the molecule on which evolution acts, and the evolution acts on what they call as the semantides or semantophoretic molecules. These are information molecules, essentially DNA and protein sequences. Protein sequencing was in place, and nucleotide sequencing was starting to come in some very rudimentary form. And then they even had the insight at this point to say that, okay, using DNA will be much better than protein, because DNA, because of the wobble base, will have a higher resolution in building phylogenies than protein sequences. So this is an important paper that came out, and Carl Woese was the first person to sort of do this in practice using ribosomal rRNA sequence. And then he said, well, prokaryotes and eukaryotes, that definition, well, it's a cytological thing. You can see it as cell properties, but it is not phylogenetic, because the phylogenetic tree is not quite bipartite, but instead it is tripartite. There are bacteria, there are eukaryotes, yes, and then what were thought to be bacteria are not bacteria, but they are archaebacteria, or archaea as we call them now. The term bacteria is not used in archaebacteria anymore, so they are archaea, and then there are bacteria and archaea, and then they say this archaea, though they are prokaryotes on a phylogenetic tree, seem to be as distant from eubacteria as they are from eukaryotes. This has been revised quite a bit, and one idea has it that the eukaryotes are really some branch that comes out of archaea, and in which case we go back to a bipartite tree, but not a bipartite tree that is defined by prokaryote-eukaryote distinction. Right. So now we have some idea of what bacteria are. They are prokaryotes, and they have their own space in the phylogenetic tree of life, and they are quite distant from eukaryotes, and so on. So that's something that we do know. Now we've come a long way. This is now it's 50, no. Yeah, this is a review. So this is about, now we are 50 years, okay, 50 years from there, close to 50 years from there. We have a large number of bacterial genome sequences out there. So sometime in January, when you look at the NCBS, there is this many bacterial genomes available, and a more dedicated bacterial taxonomy database has a very large number of bacterial genomes somewhere there, and which covers some 1 lakh different bacterial species. So very large numbers of bacterial genomes seem to be available to us today. What have we learned from all these genomes? The first genome was sequenced 30 years ago, 1995-96, and since then, this large numbers have been sequenced. Not all of these genomes are fully sequenced, so to say, but are in various states of completion, especially with these deep sequencing technologies make it easy to get to, say, 95% complete genome, but not quite the 100%. Most of those genomes that we have today are in that category. Fully sequenced genomes mean the few thousands is what we still are looking at. What we've really appreciated is the genetic range and plasticity of bacteria. We've always now known for the 100 years plus that they're metabolically and phenotypically very plastic and very adaptable. But now we've also learned that even at the genetic level, they can be very variable. And let's just start with something very simple, just how big is the genome. So the smallest bacterial genome is, I think, 100 kilobases or something like that, and it's not quite a very healthy bacterium. It doesn't have even the minimum requirements of cellular life. It's probably on the way to getting degraded and becoming part of the host cell, like a mitochondrion or an organelle of that kind. And the largest bacterial genome is the 12 to 13 MB in length. And this corresponds to bacterial genomes with as few as 100 genes, all the way to some 12,000 plus plus genes. And in these bacterial genomes, we also see that as the bacterial genome grows in size, the number of genes, and we can in bacteria think about the number of genes as a direct representation of the bacterium's capabilities, its ability to inhabit a diversity of habitats, its ability to tolerate a whole range of insults, ability to consume a whole different set of metabolic nutrients and so on, and which to some may translate into an organism's complexity, which can be defined in many ways. Here maybe the gene count translates to its complexity. As the genome length increases, gene count increases linearly, and thus its complexity and its set of abilities. And this is true for both these clades of organisms, but not so for higher eukaryotes. And higher eukaryotes is where you sort of run into what is called the C-value paradox, where you have genome size ranging 4 orders of magnitude or something like that, whereas the number of genes is pretty highly compressed and is a couple of folds difference. And we will not get into why that is the case, but except that in bacteria these are really convenient genomes to work with. I have genomes, I have gene. If I'm interested in studying genes and their role in doing whatever it is that I'm interested in, then bacteria are excellent systems. So genome size is one thing, then gene content can be something else in a way. So this is so let's say that I have a set of genes, because these are all evolutionarily related. They started off with somewhere and then they've been branching around and doing various things, and I have a set of gene functions that evolution has discovered. Now I take this set of gene functions and I ask which of them are sort of common across all or most bacteria, and which of those are highly specific to some bacteria or the other. If most of these genes are highly specific to some lineages, then it means that I have a high diversity in terms of gene functions across bacteria, and the reverse if most gene functions are highly conserved. And here you see that there are some gene functions which are very highly conserved. These would be core gene functions like the central dogma, without which cellular life cannot really exist, and that's there. And then but mostly these gene functions are very unique to some bacterial species and not present anywhere else. So that makes each bacterial species highly unique in terms of what capabilities it has. And if I believe the number 1 billion bacterial species on this number, then I really have that many different sets of unique traits. And okay, we'll say no, of course this why should I be surprised, because this is occupying a very large phylogenetic space, so I would expect quite a lot of variation as well. But it turns out even within what I call as a bacterial species, the phenotypic diversity can be great. And for example, this is E. coli, very famous bacterium, and there's a list of habitats in which this organism is found pretty much everywhere, and this translates to extraordinary genetic diversity as well. So here, number of genomes are all genomes of the same species, all genomes of E. coli. I have thousands of genomes of E. coli available today, and this is the number of genes that I find in so many bacterial genomes. And I have two shapes, one that is increasing, ever increasing sort of, it's not yet saturated, and then one which seems to be ever decreasing, and this is called the pan, this is called the core, right? The core genome is the set of genes that are found in all of these genomes. So if I'm looking at 1,000 genomes, then these genes are found in all 1,000 E. coli. I start with one and go on. Of course, after everywhere this is essentially a distribution, because it depends on what sample of genomes that I'm looking at, but I guess this is the mean or the median that has been plotted here. And then this is the pan genome, this is the total number of genes that are found across. So this is essentially, if I consider each of these genomes as a set, and the set being the list of genes that are found in that genome, this is the union and this is the intersection. The union is ever expanding. That tells me that even after sequencing a few thousand E. coli genomes, if I sequence another E. coli genome, I will find a set of genes, say some tens of genes in this genome, which has not been seen in any other E. coli so far that I've sequenced. And every time I sequence a new E. coli, the core genome is probably going to drop a bit more. So, and this also tells me something. Genomes are going to shrink and they're going to expand. And what are the processes that are involved in this? So, shrinking genomes, let's start with that. Now I think it's reasonably well accepted that in most bacterial lineages, the dominant event is DNA loss. DNA loss happens at a fairly high rate, but genomes just don't end up going down to nothing because there are sporadic bursts of gain. So gene loss keeps happening at a fairly high rate in, and this is, you can look at it, there's a lot of detail in that, but we'll not get into that. And one consequence of this sort of gene loss is the creation of endosymbiotic organisms, which sort of eventually can become organelles, for example, mitochondria. So here it's very, very clear. So I have this species this is Buchnera. These Buchnera are endosymbionts that live inside certain specialized cells in insects called aphids, and these aphids eat xylem and phloem, and which are nutritionally very poor. So these Buchnera are essentially machines that produce amino acids that and they supply to the host, and these are quite closely related to E. coli and other enterobacteria. Okay, so here are E. coli and other enterobacteria, and here are Buchnera, and I go back in time, and somewhere here is the common ancestor of both of these organisms. Now, if I'm assuming that genome size is something that reflects the sort of the branch length from the common ancestor, I'll immediately see that the common ancestor of E. coli and Buchnera looks a little closer to E. coli, much closer to E. coli than to Buchnera. Now, E. coli is a 5,000 gene genome, whereas Buchnera is a 500 genome, and therefore the common ancestor of all these is probably much closer to having 5,000 genes than 500, and that means that the creation of this particular species has involved extensive DNA loss. Essentially, this is things that are living inside host cells, no real competition with anything else, and natural selection is relatively weak in these organisms. So anything that doesn't kill can be lost from the population, and that seems to have been happening in these organisms. And similarly, you see this in Rickettsia, which are among the closest extant bacterial relatives of the mitochondria, for example, already quite degraded, cause disease as well, and also chlamydia. There are various such examples of these kinds of genome DNA loss events leading to the formation of highly crippled parasitic, symbiotic or endosymbiotic organisms. And some of these can also create beautiful jigsaw puzzles. So here is this example of two bacterial endosymbionts together living in one host cell. So this guy, the guy at the bottom, is essentially an amino acid factory, and this one is a vitamin factory. The vitamin factory doesn't produce amino acids except one amino acid, which this one doesn't produce, and this doesn't produce vitamins. And they sort of exchange, and these exchanges seem to have gone to an extent that there are certain combinations of such endosymbionts in various organisms where even something like the component of oxidative phosphorylation necessary for respiration may not be complete and still somehow shared. And in some examples, you can see heavily nested structures. There is a big eukaryotic cell inside which one bacterial cell lives, and inside which another bacterial cell lives. So you get these really tight complementary jigsaw puzzles that get formed during these gene loss events. Okay, so that's setting aside shrinking genomes. Look, talk about genomes that are growing in size. And we know now that bacterial genomes usually grow in size by the phenomenon of horizontal gene transfer. Horizontal gene transfer, also called as lateral gene transfer, is a phenomenon by which an organism is not getting its DNA from its parent, right, which is vertical gene transfer, but is getting DNA from the environment and from its colleagues living pretty much anywhere in the phylogenetic space, as long as they are living close by, and one can throw out DNA and the other can take up the DNA. This is possible, and estimates suggest that in these various organisms, this very large fraction of gene family expansions might have been caused by horizontal gene transfer, and these been associated with various interesting functions. For example, virulence. This is a brilliant classic example of Vibrio cholerae acquiring the cholera toxin through a bacteriophage, a filamentous bacteriophage. And this is also a curious filamentous bacteriophages are interesting in that they don't have to necessarily kill the host as well. They sort of replicate in the host and they sort of leak out and go and do what they do. And the cholera toxin is part of the DNA of this bacteriophage, and that transferred it into the host. And these kinds of horizontal gene transfer events seem to have been responsible for various landmark events in the history of cholera. So here is the cholera pandemic, 7th pandemic, and over time the years are marked. Here different events have happened. This involves first getting a bacteriophage which codes for the receptor for the bacteriophage that has the cholera toxin. First you have to get the receptor, and then you get the cholera toxin coming in, and then the cholera toxin can undergo various changes to form different varieties of cholera pathogen, and this can involve horizontal gene transfer events as well. Also, a very famous example of horizontal gene transfer is antimicrobial resistance, especially in the so-called escape pathogens like E. coli, Klebsiella, Acinetobacter. Not so much in things like Mycobacterium, where mutation processes confer resistance. Here you can see a nice example of horizontal acquisition of resistance or horizontal transfer of resistance from one organism to another being much higher for organisms of the same individual's microbiome as opposed to across individual's microbiome. And not just virulence and antimicrobial resistance, even fundamental metabolic processes, for example, photosynthetic components can be horizontally expanded and transferred. Now, if horizontal gene transfer is very common, is this a challenge to phylogeny? How do I define a species if genes can travel from one species to another? It turns out that, so here I can look at still core genes like central dogma genes, which are highly conserved, and those genes seem to show very little signs of horizontal transfer. Do other types of genes show that? It seems to depend on who you ask. Some people say they don't, some people say they do. It depends on their methods and how they root the tree. These are all kinds of murky areas. So whether there is HGT or no HGT, signatures of vertical inheritance, especially for the core genes like those involved in translation and so on, seem to be intact. So there is still a good basis to believe in some kind of species definition based on a nice tree-like structure. So gene content diversity is driven by gene gain and loss processes. And gene sequence diversity, this is stuff that we all sort of learn first, that you can have starting from somewhere, mutations will happen, diversification will happen. So I start from somewhere here, then different types of phenotypes emerge over time. And this is an example of something that we've done where you keep E. coli in stationary phase for very long time. And back in the late 90s, Roberto Colter's lab showed that when you do that, you have new bacterial traits emerging within that population, new mutations emerging. And you can see that over time, these are number of days that they maintain in stationary phase, the number of mutations also increases, and the genetic diversity of the population also starts to increase over time. And mutations can also produce antibiotic resistance. They may not be so important in escape pathogens where the dominance of horizontal transfer is great, but in Mycobacterium tuberculosis you have various antibiotics and mutations in certain genes showing up to confer a strong resistance to these antibiotics. And there is also the idea that mutations are random and then selection selects, but mutations need not be random. They can happen in some non-random ways, especially in terms of where they are on the chromosome, also in terms of when they occur. And even if you have mutations occurring randomly, the nature of the genetic code is such that the consequence of that need not necessarily be random. So that's a good computational exercise. Yeah, that's a good one. Right. So bacteria are very highly adaptive organisms. Both mutations and gene flux, that is gene gain and loss, determine evolutionary trajectories in some complex way. And didn't really talk much about this, but the bacterial genome is very, very heavily driven by selection. So finally, I'll talk a little bit about the questions that we are asking in the lab and how this ties in with various things that we've talked about. So talked about this. This is the stationary phase adaptation that I just mentioned. If I look at and here you see some mutations coming up, some mutations going down at the individual mutation level. And if I look at the kinds of functions that are represented by these the kinds of functions that are represented in these things, you will see that there is generally an enrichment of genes involved in DNA regulation. The color doesn't come up here. So this is the list of all the genes which are mutated at some point in these kinds of stationary phase experiment that we've done, which is only for 1 month, as others have done it for years. The ones that are marked in blue, which is quite a large number, if you can see the blue, well and good, I can't, and plus a few more are all involved in regulation, and this is an overrepresentation compared to what you would expect. Now talking about gene expression and its control, transcription is what we are looking at in bacteria when we are really, unless I say otherwise. Right? You have the RNA polymerase holoenzyme, which binds upstream of genes, and and that specificity is given by the so-called sigma factor. And once it binds to these promoter regions upstream of genes, then it can start transcription and produce mRNA. And this process is tightly regulated, and you have a bunch of proteins which are called transcription factors, and these proteins can bind into different locations relative to where the promoter is, and by being there it can activate or repress transcription by one of different several mechanisms. And there are many transcription factors, and in E. coli this transcription factor forms a nice looking, well, nice or ugly looking woolly ball network where have hundreds of transcription factors, thousands of genes. Some transcription factors regulate some genes, and you can make some pictures of this kind for what they're worth. And different parts of these kinds of regulatory networks can be activated under different conditions, therefore suggesting that different subsets of transcription factors may drive physiological adaptation, not genetic adaptation, physiological adjustment, so to say, to changes in environment. Now if you look at the numbers of transcription factors, it's kind of curious in bacteria. As genomes grow, things like ribosome remains the same. All bacterial ribosomes are more or less same. So the number of genes involved in forming the ribosome is sort of a flat horizontal thing. And as bacterial genomes grow, the number of metabolic genes increase linearly. Say some 25-30% of any bacterial genome is expected to be involved in metabolism. But regulatory proteins go in a fairly close to quadratic manner. As genome size increases, the number of regulatory proteins increases in a superlinear fashion. So I have organisms, these endosymbionts don't have many transcription factors, and transcription organisms somewhere in this size range can be have a lot of transcription factors. So this is what we are asking. So we have all this information. How do transcription regulatory networks evolve, and more specifically, how do transcription factors evolve? So this is where the kinds of questions that we are asking. So cutting a very, very long story short, I have transcription factors present in some numbers that we saw in that graph. I can do some phylogenetics and sort of try and infer what might have been present in the last common bacterial ancestor, last common archaeal ancestor, and last universal common ancestor, which is a highly debatable subject where this thing has to sit. But we make some assumption and see where they happen, and clearly these are the numbers of transcription factors that are present. Very, very small numbers, right? If you go back in time, these organisms have very, very little regulation going on. And a good fraction of these regulatory proteins are the so-called basal transcription factors, which are involved in the core RNA polymerase machinery itself. And the other transcription factors that are involved seem to be involved in very, very selected functions. For example, DNA metabolism. One of the regulators that we can find in these common ancestors is the one that is involved in regulating ribonucleotide reductase, which is involved in producing deoxyribose nucleotides from ribose RNA. So that and which is central to transitioning from an RNA life to a DNA life. This already has DNA life. This is already a complex organism that we are talking about, and it's kind of curious that it might have already been regulated at that time in response to ATP or something like that. And then we have alternate respiration pathways seem to be regulated this time, and also metal response. For some reason, I don't understand that geology very well. It looks like these common ancestors had a large number of proteins that are stabilized by metals, and metal response and seems to also be very, very commonly found in these organisms. So how did transcription factors emerge in the first place? So there is this model. So why is this question also interesting? Though transcription is necessary for a DNA-based life, its fine regulation by these transcription factors is not fundamental. Endosymbionts that we talked about may have zero transcription factors, but they can still do some kind of regulation based on energy sensing, supercoiling, something can happen. They can still do broad levels of regulation, but this very fine regulation by these transcription factors seems to be non-essential or non-fundamental to life. To a minimal life. However, things like DNA compaction may have been fairly fundamental, because even if I have a minimal organism with some 100 genes, it will need to sort of compact its DNA by 2 orders of magnitude or something like that to fit inside any reasonable small cell. So Sandhya Visvesvaraya at IISc and Steve Busby at the University of Birmingham came up with this intuitive proposal, which they wrote up in a review, where they said, okay, these non-specific DNA binding proteins, which are probably involved in shaping the DNA or compacting it, are ancestral proteins on which certain traits that are required for a transcription factor may be evolved. And these traits can be like recognizing certain DNA motifs specifically, or sensing some signal, and or if it's an activator, finding an interface that will recruit the RNA polymerase. All of these might have evolved on top of this. So is there what is the evidence to search for this? One such possible evidence is the manner in which sequences are organized. This is also a very important thing that we should teach students, but it's not really done. So there is a hierarchy in terms of how sequences are organized. I have a sequence. Sequences can then become part of gene families which are fairly closely related at the sequence level and may share functions at a certain resolution as well. And then at the next level are superfamilies, where the sequence level similarity could be weak, but structural similarity is much higher, but the sequence similarity is strong enough for us to assume a divergent evolution from a common ancestor. And then at the next level are this very small number of folds where I may have literally no sequence conservation, but you could still have a broad structure, and that probably came from convergent evolution. Now, when I look at this, if I look at superfamilies that include transcription factors, you also find that other kinds of DNA binding functions are included as part of the same superfamily. So here are things that are involved in transcription regulation, but then you also see things like chromatin binding and other activities for part of the same function. So if these have a common ancestor, then what is the evolutionary trajectory of these pathways? So it's not easy to do, because it's not easy to do alignments where at that resolution to be able to get nice phylogenetic trees, and you also should have some information on what is a transcription factor, what is not, etc., etc. Not straightforward. So the candidates on which we can do this is fairly limited. I'll just give one story here. That's these two proteins, and this is an interesting photo that one of my colleagues, Mukund, sent me when he knew that I was doing this work. And then I looked at the subject and thought he had some point to mention, and then there was this photograph which he had captured somewhere in Bengaluru. There is that bike which has HU in the number plate, and here is one which is 1HF or IHF on the number plate. I don't know how he managed to spot this in a traffic signal or wherever it is in Bangalore, but he did that, and I'm using this everywhere. So these two proteins, here is HU, here is IHF. HU binds to DNA non-specifically, and IHF binds to DNA specifically and bends the DNA, and does has some regulatory roles. Not get into that aspect. It's enough to know that this is non-specific binder, this is a specific DNA binder, and most importantly, they form part of the same sequence family-ish superfamily, and they can be together very, very nicely aligned. Which means if I can make a nice sequence alignment, then I can look at its phylogeny. And now I can, and if I do that, just basic sequence clustering, basic bioinformatics will tell me that IHF has two subunits, HU also has 2 subunits. The 2 IHF subunits in yellow and red are separate, and the HU subunits are 2 HU subunits sort of intermingle, and they are separate from the 2 IHF subunits. This is basically a more classical tree-like structure where you see all HU sitting here, the 2 IHFs separate. The pink one is an IHF-like thing, ignore. And now I can look at these proteins, their presence and absence on the species tree. This is not a gene tree. This is the species tree of bacteria, and species trees need not always match all gene trees, and that's something to always note. And the blue dots here shows that there is an HU present or inferred to be present in an ancestral node, because I have data only for the leaves, extant organisms. For all of these I do not, so I make some inferences. And HU probably present in the last common ancestor, and then lost in some lineages, but very highly conserved. IHFs came much, much later. So if I start here, they came somewhere here. So this suggests that HU is ancestral to IHF. Now if I build a gene tree, now I can make a tree, but usually when I make the tree gives me relationships, but it doesn't give me a direction. Any of those leaves could be the starting point. So to find out what is the most likely starting point, I have to find ways to root the phylogenetic tree, which it's a pain, but it can be done, and in this case we managed to do some kind of a rooting of this tree using what's called an outgroup, and that tells me that the rooted phylogenetic tree looks something like this. I start from here, all the blue ones are HU, and the red and the yellow ones are IHF. So starting from here, I start seeing HUs coming first, and then the IHFs coming later. So the HU seems to be closest to my ancestor that I'm seeing. And if I can reconstruct these states, I can also reconstruct sort of sequences with some degree of confidence, of course, and that again tells me that the last common ancestor is going to look a little bit like HU. And this is consistent with what I saw in the species tree, and these are 2 very independent sources of information. And again, what this shows is that HU and IHF are not like 2 separate branching things, but IHF emerged from a late branch of HU. So HU had already diversified, and at some point, out of some branch of HU, the IHFs emerge. And and IHF seem to have evolved to become a transcription factor, gaining sequence specificity in different ways. IHF alpha or IHF-A seems to have co-opted residues already present in HU towards becoming what it is, and IHF-B employs different set of residues, and these two have to come together to form a heterodimer to become a transcription factor. Homodimers of these at least are not known in E. coli. So this is what comes out of this. Basically, a whole bunch of sequence analysis that can be done. This is not too complicated, can be done in master's undergraduate level type thing, and in fact this was done by a master's student in the lab. So says that HU emerged very, very early, the tree, and then diversified, and then somewhere here one of the IHFs came, and then that diversified into two different clades, and then along one of those clades the other subunit of IHF came, and then that went somewhere like that, and then there's probably a horizontal transfer event from this into this to create that IHF alpha like that I talked about, the pink one that I asked you to ignore, but that's there. And then though most organisms have only a single subunit of HU, E. coli and related organisms, which is sitting here somewhere, contain two subunits of HU, and both homodimers and heterodimers are known and with the different functions. And the suggestion at this point is that the second subunit probably came from horizontal transfer in this ancestor somewhere here. So this is all in one sense 101 bioinformatics that can be done, and doing it well is no longer 101, but can be done in at some level to probably you'll get similar results anyway. And what you'll find is that the ancestral protein was HU-like, and a transcription factor that has evolved from a non-specific DNA binding protein in a certain manner. So that's pretty much what I wanted to say. The bacteria are highly plastic metabolically, and they're genetically very, very diverse, and they evolve in different ways, and their transcription network has also evolved from something very, very simple in the last common ancestors into various diverse structures. And one route towards making a transcription factors appears to be starting from a non-specific DNA binding protein and finding some interesting ways to form a sequence-specific DNA binding transcription factor. And forming a transcription factor essentially means that you've gained an ability to regulate a certain response of the cell to some conditions, and that can come from not being able to do so in an ancestral state. And a lot of people who've been in the lab for substantial periods of time, various collaborators and various people who've given sage advice at various times of my career, funding stuff. Thank you very much.
Thank you, Aswin. We'll take any questions. Great. So maybe I'll just get us started. So I think a lot of what, I think you shared several very cool stories. I really like the HU-IFP one. But I think that a lot of the representation is probably very new to the crowd here. So for those who were kind of focusing on the names, and this is, I think, something that comes up in textbooks a lot, that biology often overemphasizes taxonomy, right? Without us having to illustrate exactly why it's important to study that. So could you just underscore the essence of what one learns from these studies in maybe slightly non-technical language?
Sure.
Yeah, thanks.
So the first idea that comes here is the idea of how we classify things, right? And phylogeny differs from any classification, though it looks like classification at the end of the day, in that underlying the phylogeny is the assumption of a process, right? The process of certain rates of change, right? Starting from something to the other. And then I have, eventually I make some sort of a map, which is basically a binary tree, right? And then this so bifurcating tree. So finally, when I have this information, then I can do a whole bunch of things with it. And there are different types of trees. One, there is the species tree, which tells me that I've started with an ancestral organism, and from which I've split in various things, and various descendants have come, and I have some set of organisms that exist now, and I know where their ancestors are with a certain degree of confidence. And then I have now, okay, so now once I have a species tree, I can trace the evolutionary history of anything that I have information on for extant organisms. Now, if I put in extant organisms, then I can see what sort of things would best fit my current distribution of data, and I said, okay, I might have started off from here, and then over time X, Y, Z changes have happened, leading to what I have at the moment. So for a certain trait that I am looking at, I have some inference on what its trajectory would be. And if I assume some sort of this bifurcation keeps going on forever, maybe I can also, predict what's going to happen in the future. And the confidence of these things increases quite a bit if my phylogeny is shallow and dense. For example, COVID-19. Right? Huge numbers of genomes for a couple of years of data. Very detailed phylogenetic information is available. And I understand that even some certain viral vaccines use this kind of information to sort of make the next year's vaccine, make these kinds of predictions. So the trait that I'm looking at can be phenotype, right, which is again a few steps away. It can also be genotype. A gene tree need not necessarily track the species tree. Species have their own life, genes have their own thing, and they can do their own funny thing. So if I make a gene tree, on that gene tree I can make certain other inferences. I can sort of try and see what the ancestral sequence looked like. So for example, if I'm doing something like this for an enzyme, I can try and see, okay, did this enzyme have the ability to perform the same reaction, for example, in the past, or did it suddenly gain this in some lineages, or has there been some ancestral enzyme with certain properties which is diversified and become two different enzymes. So how did all this come about? For this, I can make data-driven inferences based on these kinds of trees, and the representation here is essentially trees that keeps going, on which I'm making inferences. That's pretty much it. Conceptually, very easy, but actually building it was really difficult, because there is no one tree that works. Lots of trees, each of which is a hypothesis, and some trees fit the data better than the others. I have to sample a lot of things, and being able to do that has really become possible only with massive increase in computing power that's happened more recently. The kinds of regulatory network evolution people were trying to do 20 years ago, but it just wasn't possible to do it at this phylogenetic depth at that time. And most people, probably except me and maybe a couple of others, have lost interest in that problem over the years, and I still retain that interest, so I'm able to do that, revisit that problem now with the backing of a lot of phylogenetic information.
No, I'm quite unfamiliar with this topic, but I just wanted to know if all these details of evolution of genes and the proteins, etc. I mean, did all this was it done on the database or is it data plus experiment or in both?
So this is purely data-based. There is this is purely data-based, and inference is based on the data. And in some cases, I'm not very confident it's possible here, but in some cases where the sequence similarity is quite high, it may be even possible to take from here and try and synthesize ancestral enzymes and try to characterize their properties experimentally. So that has been done in some cases.
Okay, another thing, again as a layperson, I mean, so what is the new insight that, earlier from popular books, one knows gene mutation and, gene sequences differ and therefore, and one also knew that for its expression, some proteins are, some mechanisms are needed. So is the new insight that these mechanisms themselves are evolving as the G?
Yeah, that's true. So mechanisms are, even at the very detailed level, if we go back, if we go even forward, then one can also see that even if I have the same transcription factor, that might have evolved different functions in even closely related organisms, and one sees, what context this might have
So what is the, this is just detailing. What I'm saying is it looked like a general insight is already there, but you are detailing it more and more. Are you getting some new insights?
So the question is, at what level of detail is one interested in? So if I'm going to say that mutations exist, and that they happen, and pretty much everything is at the level of mutation. But just saying that mutation exists is also not super useful. And there are so many different processes, and they have their own life, and how a certain process has evolved over time is probably different in different cases.
Is there some predictable? Can you control environments and say, okay, well, it will go this way, or that's not?
In some cases, yes. In some cases, even experimental stuff has shown that you have very strong parallelism. Is it predictable? I don't know. But when I see one example, then I know that this is likely that I will see similar things in different pathways.
Okay,
but a priori, I don't know.
Okay, my last question is, what kind of abilities would a BSc bio student, what kind of prerequisites are needed for this kind of?
So a little bit of programming knowledge will help.
Okay.
But I do have some students who come with no programming knowledge, and by just Google and then finding some commands and so on, they're able to do this as well. And otherwise, some basic appreciation of protein sequences. And what or gene sequences, what they look like and how they are organized, which when I was studying in college, this was sort of given a lot of importance. But now when I interview newer students, I think they sort of give that the short shrift these days, I think. But having that information is useful.
Thank you.
Hi Ashwin, nice talk. One thing which struck me was you said that mutations are not random. Should I take that to mean that mutations are not randomly located along a gene? They could be some specific areas which are more susceptible to mutations. But to me, a sort of definition of randomness was more about that a particular mutation does not, the randomness is because does it confer any deleterious or beneficial or does nothing at all? That region could just be not expressing anything. So for the next generation, is it going to have anything? Is that still random, or is it even, can it be not random in that? I can understand it's not random in location. There might be some locations which are more prone to having mutations.
Yeah. So it's both. Especially recently, Deepa's lab in NCBS, they published this paper where they showed that the process is as random as we can create in the lab, but then under certain circumstances, you're basically producing not neutral mutations. You produce not deleterious either. You're actually producing advantageous mutations. So yeah, that level of randomness, maybe we've been taking it for granted without enough justification.
Any more questions? I think let's thank Aswin for nice talk.
Thank you.
There's tea outside. Oh, there is one. Sorry.
Sorry, since nobody else is asking, I'm asking. And so I'm an astronomer, and I believe you are an ex-physicist, or at least
No.
Okay.
Never
Well,
came.
whatever. So suppose, from what you said, something of a bit of a worry comes into my mind. Suppose I'm trying to look at other civilizations, other, not necessarily civilizations, other forms of life elsewhere in the universe. And from what you said or what you imply about the field, I don't know whether it's your work or some, it's actually leads to a great deal of frustration, because you are implying that much of the present day phylogenetics or organisms who have that kind of is because of not vertical transfer of genes, but because of horizontal transfer of, which is highly dependent upon your environment. So if we have evolved on planet Earth up to wherever we are, and that's primarily or very largely because of horizontal transfer of information, it will be very difficult to find something like this, or even something of what you call life elsewhere. So because your implication seems to be that much of the things is happening because of, not because of my parents, but because of my neighbors.
Especially for bacteria.
Yeah.
Especially for bacteria, right?
But, I mean, we have evolved from bacteria.
Yeah. So it's but probably, I guess, the origins of life is probably not because of horizontal transfer, I guess. It did have a vertical component. I guess now horizontal transfer is kind of dominant primarily because it allows, instead of, if I'm just doing a vertical thing by mutation and so on, if I have to gain a new ability, it's going to take forever. But this pathway essentially gives the bacterium the ability to gain a whole new function in one go. This whole bunch of DNA gets transferred into to that. And that's probably helped in the dominance of bacteria on the planet today. In the absence of horizontal transfer, or to a small amount of horizontal transfer, one thing that it's possible is that bacterial DNA would have shrunk to very, very small things, because in the absence of these gain events, DNA just keeps shrinking until it would probably get to a point where it's just a minimal selection. I think that's going on. And assuming that's not the case, and then maybe you'll still have bacterial life, but probably just not as versatile and as dominant as they are today. The classical example is antibiotic resistance. Antibiotic usage has gone up, and resistance has also gone up concomitantly in the last 20-30 years. It just exploded. Maybe that wouldn't have happened. Bacteria still existed, found some ways to tolerate things, but that property which helps it to adapt uniquely to the circumstance that's present on planet Earth now, maybe that ability would not have evolved so fast. So life, I don't think it really matters to life per se, but to the dominance of this organism today, yes.
All right. Thank you.
Thank you.
Again.